Daddy is cute, Daddy is strong, Daddy is brave, and Daddy will not stop.
About 3 to 5 meters. So.
Okay. So I think we're live now. And let me move some things around while I'm still trying to get the...
Well, I'm going to just say a quick hi, and then I'm going to mute myself.
Okay. You don't want to have the conversation?
Hi. No, I do. I just have other noises in the house, and while you're getting started, I'm going to sit quietly, and then I'll help with chat and participate. I just need a good morning, and then I'm so excited. Voice.
Okay, so one thing you could do is just, while you're muted, is check to see that the sound for Studio and I are both coming through on the stream side. Can you talk a little bit, Studio?
Yeah, good morning. Hi, I'm here on the stream today, and we're looking at samples that I collected from June Lake in the Sierra Nevadas in Mono County, California. So.
Okay. Well, Access Deny says that the sound is coming through for everybody, so that's good. And swell. Thank you, Access. And hopefully everybody's at the right volume. I can make Shelly a little louder. I don't know if you want me to call you Studio or Shelly, or what do you want me to call you?
You can call me Shelly.
Okay.
I very rarely in my
All right. So this is oops, now I'm hearing myself somehow. Is that coming through on your side? Right. So oh, it's here probably. Hang on. I gotta find where my sound's coming in and tell it to stop. Okay. All right, so we have some samples that Shelley collected for us, and we should start by maybe giving you a quick introduction. You are a scientist and an artist, and PacificPlankton has provided a link for you, and and do you want to talk a little bit about the kind of stuff you do? I know you sort of do some pigment extractions from algae, and I've seen a bunch of your artwork on Instagram. So there's a link in the chat for people who want to check that out, and we can probably put some of that up if you have some favorites you want me to try to stick up on the screen.
Well, yeah, so I am in my day job, I am a software designer. So watching the SEM software is always an exercise in patience for me because scientific software is just a universally difficult to use, super small, kind of drives me nuts. But in my artistic life, I primarily work with cyanobacteria and microalgae. And I have several kind of projects that I'm doing right now. One of them, pardon me, is extracting pigments from microalgae, more specifically cyanobacteria. I have a huge interest in harmful algae blooms and cyanobacteria. So I've been practicing with both algae that I grow on my algae farm. So I have several different kinds of blue-green algae or cyanobacteria, as well as different microalgae growing here in my house. As well as what I will do is go out to harmful algae blooms in my community. I'm based in Seattle, Washington, and will collect algae from those blooms and extract those. So the kind of concept around there is kind of understanding the color of a place and color of a specific natural phenomenon. So also related to the samples that you're seeing today, I have been referencing a historic collection called the Ehrenberg Collection, which is one of the foundational collection of specifically diatoms, but really microorganisms across the board from Dr. Gottfried Ehrenberg, which is currently housed in the Natural History Museum of Berlin. And I've been going to various kind of historical locations on the west coast of the United States to sample and compare the samples of what you find today to what was observed in the 1840s and 50s. Much of the samples that were collected were related to kind of exploration around the gold rush, and that kind of brings us to June Lake here. So June Lake in Mono County, which is part of, really kind of tells the story of the western United States landscape expansion, as well as the land use, and how there's been very hot debate on what should be, how this land should be used. So yeah, and I'll pick out some various art pieces to show, but that's kind of what I've been working on now. But generally, I will actually look at water specifically, but sometimes soil samples, and reflect upon the what's found there, and kind of the idea that it's defining a sense of place. So yeah.
Okay, so I want to just back up for a second. You have a algae farm inside your house?
I do, yeah. There are several 1-gallon containers that are continuously aerated. They live on a sprouting mat, which will keep the kind of temperature of the water in between. I'm a Fahrenheit person, so apologies. I don't convert to Celsius very well in my brain yet. But generally between 78 and like 86 degrees Fahrenheit. And I also have 12 hours of mixed wavelength grow lights on them as well. So yeah, I farm various different kinds of algae, specifically species that are named for their pigmentation. So right now I have Anabaena and Spirulina, which are two cyanobacteria, which attract like a blue kind of purple color. And in the I also have Hemiococcus. That's probably a terrible butchering of the name. You might know it as things that grow in your bird bath when the water gets low. It's that pink scum. You'll see it in fountains and bird baths. It has a red accessory pigment, which is commonly used in cosmetics. You can't see my screen, but I'm actually wearing cosmetics that specifically use algae pigments today. So I also have a kind of and I have a Daniela salina, which is a extremophile. It grows in salt ponds for Pacific plankton. The salt catchments outside San Francisco Bay are a great place to go view them. They extract beta carotene. They actually have the highest level of beta carotene of any organism. So they're farmed for beta carotene supplementation. So, yeah.
That is super cool. And also, you're married, right? So your husband also has agreed to having a giant algae farm in the house.
Yes, he thinks it's cool, and everybody who comes by thinks it's cool too. I don't eat my samples because I compliment everyone who uses algae supplements or those kinds of things to make sure that you get them from a reputable source, because I don't have good FDA style controls for my algae. So I like, for example, spirulina, for whatever reason, is my lab weed. It is everywhere. I find it in everything. Like if I leave even my freshwater samples open for any period of time, I have spirulina in them. I don't know why. Maybe it's my cross contamination, but yeah.
That's kind of interesting. So, so everything that you have is sort of contained, though, right? Like, so they're in sealed containers. So how are they contaminating?
Well, they're kind of semi-sealed. So I actually I have the lid. I have the lids, but they're not like airtight, and they're
They gotta breathe.
continuously too. They're also next to each other. So and this particular space that I'm in is like my studio, so it's not huge. I also believe that probably I cause a lot of the cross-contamination because I don't have like very strict algae protocols. So it's not something that I necessarily need to emphasize.
Yeah. Oh, anyway, so that's really cool. And then you have specific algae and cyanobacteria that you are cultivating for particular pigments and getting particular colors from them, and then
Correct.
And then you're using those for. You said your makeup has got the pigmentation in it. Some of your makeup, is that? Did I hear that right?
I mean, it's from industrial sources, but porphyridium is a really common pigment that it's a purple, so it comes from porphyridium is a microalgae. It's a red algae, and it's one of the very few microalgae that are red algae. Generally, red algae are larger, what we would consider seaweeds. So, carrageenan is primarily derived from a red algae, as well as nori. So, nori is a rhodophyta, a red algae. So I've had a lot of success actually collecting red algae from just the local the local beaches. I have a license, like you have to get like a fishing license in Washington State to collect algae, or seaweed. So I have that, so I can get crabs and seaweed. They're the same thing for whatever reason, yeah, so I've had a lot of success in actually extracting a purple, but porphyridium is commonly used as kind of a purple agent in cosmetics, as is anaxaphene. I said that completely wrong, but it's the red accessory pigment that are in things like the helium, which is commonly found in bird baths, and in sanguina, which is the microorganism that is found in glacial snowmelt, which is known as watermelon snow or blood snow.
Oh yeah.
Mm-hmm.
That's really cool. I talk about this in my trying to think of which one of my classes. I think hydrology class. I talk a little bit about the watermelon snow, just as like a odd things that happen with snow sort of comment. And the students, every time I show those pictures, it looks like something terrible and tragic has happened in the snow. When you see it, it does look like sort of blood, right? And then when you squeeze it, the sort of bright red color comes out of the snow even more. So that usually freaks them out. But
yeah, it's definitely we call it generally like watermelon snow in the United States, because I feel like our culture tends to have like cutesier names for things. In Europe, it's known as glacier blood. I've seen it. Blood snow, or is something that I've heard in Russian content as well. So that's what I call it, is blood snow. And I've actually collected that. I've gone up to Artist Point in Mount Baker, in the Mount Baker Snow Colony National Forest, and collected the blood snow. And there are also blood snow surveys here in Washington State to kind of understand climate change, because the red pigment helps protect the algae from kind of extreme conditions like sunlight, especially on a snow field, the sunlight can be really intense. And what it does is it actually heats up. So what makes it even freakier, right, is that the places where the blood snow algae is, it melt more quickly. So they actually make gashes and wounds in the snow field. But that can also increase the melting of the snowpack. So the more of this algae is out there, the hotter the surface of the snowpack gets, and the faster it melts. So
super cool. I actually have never seen it in person. I live in Indiana. I grew up in Ohio, like you, and I think you kind of need to have like high altitude, right? It's not something that happens at like in low elevation areas frequently, and it's actually if I remember correctly, it's actually a green algae, right?
It is, yeah. And if you actually look at it, I'll share some of the images that I've taken on Instagram. It tends to also, I think, be coexistent with other types of green algae too. So it's kind of the dominant species, but there's also, and it is a green algae. A lot of those red pigments actually come from what we would call a green algae.
When it gets sort of outside of diatoms, my understanding of algae sort of shrinks very rapidly. I know most of the names of, some of the things that you brought up, like Anabana, for example, I'm familiar with, because we talk about it in class when we're talking about eutrophication, and it's a common nitrogen-fixing cyanobacteria. So like I know some of the basic green algae cyanobacteria types, but if you start to really get like into the weeds, I won't know them. But I think that also nobody cares if you get the pronunciation, right or wrong, because it's Latin. So just say it however you're going to say it.
Yeah, I mean, I'll do my best.
Some of them are sort of a mouthful anyway, so.
Yeah, I mean, they obviously have like sensical roots, like it means like red oil extracted from X or whatever, but yeah, it's like yeah, it's difficult.
I like the way Diatom's attack says, just say it with confidence, and it's the way it is.
Yeah.
I like that.
No one will stop you. It's Latin, and if they don't like the way that you pronounced it, that's too bad for them. So it's a dead language, and generally, I only get corrected from the way that I pronounce my diatoms by Europeans who don't like the way that I say Eunosha, or any number of any non-American people who don't like the way that I say something. But it's the way that most Americans will say those genus names. So like I don't care if people don't like it. They just have to deal with it. It's a Gonfanella, a little Gonfanella. A little tiny.
Okay, Gonfanella. Gonfanella, not Gonfany. I have to remember.
It's something in Olivaceum, like Olivaceum. Sorry, it's a cute little diatom.
Why are you apologizing about the cute little diatom?
I don't know. I got excited by it, I guess. It's I don't think I've seen this one in this June Lake stuff before, but I haven't spent a lot of time looking at. I have a grad student right now, Laura, who's working in June Lake, and she's actually mostly working in Gull Lake, which is right next to June, and we have sort of a long research history for the last 5 years or so. We've been sort of working with my colleague Mike McGlew from the University of Kentucky on June Lake stuff. And so for me, that's it's a really cool region that we're working in. And it's also cool to me, you said you were sort of going back through and looking at the stuff that Ehrenberg collected for diatoms. He's a sort of a giant name in diatom, taxonomy. So, and a lot of their early collections were used to sort of get a handle on North American diatoms, right? So they did these sort of expeditions where they went to all these remote places and collected material.
Yeah, this is of course actually, I had a question for you. Why are you specifically looking at kind of the Mammoth Lake area? So why are you looking at June Lake and Gull Lake and those places specifically?
So, in particular, really this started as a project from my colleague Mike, and he has been working in the Sierras for longer than I have. And he had a master's student who was interested in doing diatom work on one of their sites, and asked me if I would, help them learn the taxonomy and learn, because they didn't have any background in it. And so, so I was on their master's committee, and then they came here to the lab, and we spent some time sort of talking through the taxonomy, and they sent me samples. We processed them here and sent them back to them as slides, and so we're collaborating with them quite a bit on it. And the reason that he is looking at it, the reason that's a focus for him, is they're looking at the history of the hydroclimate for that area, and there aren't a lot of paleolimnologic records where people have looked at the hydroclimate for that in a way that is rigorous. So that they have long-term histories of where the lake levels used to be from the Pleistocene all the way to today in a way, because there's these ancient lakes that were the pluvial lake systems that were very large. And then as they contracted, as the climate started to change and the glaciers started to recede from North America, out of the last glacial maximum, all those big giant lake systems that were sort of covering states in the western part of the U.S. shrank down into little basins. And so there's a whole bunch of records from when Mono Lake was was a lot higher, and it had connected some of these lake systems together. And I think it probably wasn't called Mono Lake at the time, but whatever. It's in that basin, the Horseshoe Basin, associated with the southern part of the Mono Basin, and they're looking at really trying to figure out what the hydroclimate was like. So they're aiming to get things like snowpack and seasonality and lake level, and those are things that diatoms are really valuable at trying to providing information about. So I look a lot of the diatoms to reconstruct the climate, and that's primarily my role as a paleoecologist, is basically focusing on using diatoms to tell people, okay, well, lake level rose or lake level declined. And then in the process of doing that, I came across a whole bunch of really interesting things. One of them is that there's a bunch of endemic species in June Lake, and so things that people had observed, they probably just lumped them into something else. But when we started really looking closely at them, we realized that there's a bunch of stuff in there that's not what people thought it was. And when you have these sort of isolated regions where you have a lot of endemism, then it becomes super interesting from a taxonomic perspective and from a evolution sort of perspective. So we're working on actually developing right now some new grant proposals as well. So we've been doing everything that we've done. We've published, I think, probably 5 papers so far on the June Lake record, and we've done it without any funding. Like, everything's been done basically from start money that Mike had, or from stock material I had in my lab that we could use without getting any NSF funding. And we've gotten some big papers out of it as well. So what we really want to do is go back and sort of do everything with a little bit more detail and try to find the connections between when these lakes might have been hydrologically one system, or potentially several systems as they sort of link together in a patchwork way, and try to look at the evolution of these endemic species and where do they come from, how did they sort of reach that position where they are. And it's particularly interesting to me because when we started looking at the June Lake Stephanodiscus species, there's one in there that I described last year, Stephanodiscus coruscus, which I thought was like a sort of a coelacanth-like organism. Like it was supposed to be extinct, and then when we looked at it in the SEM, we found that actually the ultrastructure was a little bit different. It was enough that it was probably a new species, but it had only been reported as an extinct species before, and when I started looking at it, I was like, what is this? How is this alive? It hasn't been reported as living for like 1 million years. So I became very interested then, and then now we've got a lot of questions about that particular species, but also about endemism in general in that basin. And so there's sort of I have like a mixed, I'm all over the place when it comes to my science objectives, because I also have a paleoclimate side of it that I'm very interested in, but then there's this sort of diatom evolution, organismal sort of biodiversity issue that I'm sort of focused on as well in my recent research as a taxonomist.
Very cool. I'm always curious to see what draws folks to specific bodies of water. And I think for me, I camped at June Lake, and June Lake, at least in the beginning of the 20th century, has had a lot of kind of intense human interaction, although, to be clear, the Paiute folks have been there for thousands and thousands of years and have lived and utilized the resources of the Mono Basin. I specifically came into that area because of the danger that Mono Lake is facing. So Mono Lake is now at its lowest it's ever been historically. So there were, I believe it was in the 90s, maybe 1992 or 1994, when the preservation of Mona Lake was actually finalized, because many of these mammoth lakes, including June Lake and the artificial Grant Lake that is up there as well, were slated to be water sources for Los Angeles. So, and Grant Lake is still a part of that. This is something that I also experienced when I was the artist in residence at the Mojave National Preserve out in Zzyzx, California. There are a lot of very thirsty communities in Southern California and not a lot of water sources. And Mona Lake was a very hard-fought ecological kind of preservation project, and it's still not completely settled. I have a big interest in natural springs in Mona Lake, the beautiful tufas that you see that are the big stunning kind of calcification shapes that you see in Mona Lake are formed by natural springs. I actually found a natural spring at the edge of Mona Lake because the water was so low. But I was out looking for natural springs around Mona Lake because it's kind of at that place in the water table around June Lake and Mona Lake where like lots and lots of kind of fissures in the environment are there feeding into this basin. And I couldn't access any of the natural springs because they were on City of Los Angeles property or private property. And I think that that's really pretty telling of like kind of how hard fought the preservation of these areas are, in addition to the really intense human interaction that's happening. Like June Lake is a resort lake. There are multiple campgrounds. There's the city of, well, it's not a city, it's like a village. There's like a village of June Lake that has been there since probably the 1910s or so. So it's really, there's a lot of very intense kind of human activity in this region.
Yeah, the city of June Lake is sort of interesting because its location is actually right between Gull Lake and June Lake. Like it's on the sort of straddled area, highland, sort of between the two lake systems. And so it's particularly interesting for us because we have, at least our preliminary evidence suggests that sometime in the past the lakes were higher, and all of that would have been underwater, right? Or parts of it would have been underwater, and so it, it's like a city built in some ways in the bottom of a lake, right? Like if that were to rise back up to its former height, it would be, like a whole lake would have been, that city would be underwater, right? So it's kind of also interesting because the sort of anthropogenic effects that you're talking about show up in our record in the recent publications that we've had. That's become obvious that that humans have had a huge impact on that system, because the last 3,000 years that we've looked at, there were basically just two planktic species that dominated the lake system, and they just alternated with seasonality. And we saw this Stephanodiscus that's like Coruscus-like diatom, and then another probably endemic species that's like Lindavia ocellata. One of them prefers stratified conditions, and one of those prefers sort of well-mixed lake conditions. And so we can use them to give us a sense of what's going on with the water column. And it's that way for thousands of years. And then in the last 100, maybe 150 years, suddenly there's all these other plankton that show up. And a lot of them are indicators of nitrogen. So they probably are related to wind-blown atmospheric nitrogen deposition. But also some of them are just like, there's a whole bunch of variety of different types of Stephanodiscus that show up, and different types of Lindavia that show up, and they become really much more common, and the whole plankton becomes much more diverse, in particular in the last 100 years or so. and we attributed that in our paper to global warming, to industrialization, to farming, to basically human influence on these really pristine habitats. So we definitely sort of get that sense that it's played a large role in the way that the lake has developed. And I think it's really interesting because you can see how dramatically different the modern record is to what we would have observed through thousands of years of the lake history, where basically it was just the same two species, right? Occasionally there were more, but never really abundant. But the last 100 years is just like crazy how different it is.
That's really interesting that you've said that, because one of the things that the most recent artwork that I've been doing has been an exploration of the Columbia River basin. So various places along the Columbia River, where I believe the Fremont expedition of 1843 to 1844 was going along, and I believe that those samples were actually given to Dr. Ehrenberg the following year to examine. And what I've actually found is I have found more than what Dr. Ehrenberg observed, and at least recorded and sketched. And that could be, I am not studying it systematically like you all are. So looking at kind of like the longitudinal, record. But anecdotally, I've seen a lot more like Pinolaria and Ulnarians, and there are there could be a lot of reasons for that. I'm looking at a live water sample really recently collected from the area, so just like as opposed to something that might be dried on a card or something that is sedimentary. But I think that that's kind of one of the things that I'm really interested in, how our microbiome has changed. So for June Lake, something that was a relatively isolated Sierra Nevada lake that would probably only be accessible to humans certain times of the year and would probably only be utilized during those times. What does it look like when we have now like a dam and we have waterworks taking water out of the area, multiple communities, also things like stocking fish, putting in boating, so even use of things like sunscreens and things like that. So what does it actually do? I think the idea for this kind of came to me a long time ago when I'm originally, like you said, from Ohio, and there are lots of really cool cave systems, limestone cave systems in Kentucky, Ohio, West Virginia, kind of like in the Appalachians. Mammoth Cave is kind of the really famous one. But when I was touring those as a little girl, I noticed that there were kind of green algae growing on the stalagmites. And the tour guide told me, like, those weren't a part of the cave. Those actually came in with people. And I was like, interesting. Things that you just can't even see, the microbes that you bring in with you. So, and that's fascinating that you found kind of an increase in diversity, because that's kind of what I'm seeing too in the Columbia River, where we've gotten all kinds of crazy interventions, including international shipping and one of the largest agricultural basins on the planet. So lots of, as well as hydroelectric works.
Yeah, so there's actually sort of a lot in what you were saying. And so one of the things that I teach classes, obviously, and I'm a professor in my spare time when I'm not doing streaming. When I'm not moderating PacificPlankton's channel, I have a job as a professor, and one of the classes I teach is this hydrology class, and one of my favorite things to talk about is like how much environmental impact is associated with dams, and I have this really cool graphic that I sometimes show that is like it shows the aerial extent and the amount of damage that's done by different things that can happen, like a whole range of potential hydrologic events that can happen, and then in the bottom corner, basically taking up the largest part of the entire um, uh, diagram is meteorite impact, like like its damage on the environment and how big it is, and then just slightly smaller than that meteorite impact is dams. Just like in terms of like the total catastrophic effect that they have with respect to the environment, they're just slightly less damaging than a meteorite impact. So there are really huge potential consequences associated with the impoundment of systems, and
Good news,
so
everyone!
I like to sort of like point out how that fits in there. But for June Lake, even they started stocking it with fish as well, and that was one of the things that we looked at in that paper that PacificPlankton has linked in the text there in the chat. And I think at some point maybe I posted that paper or a link to a free version of it. I think that one actually might be free. I think you can just get it and download it. But we talk a little bit about the introduction of fish and whether that's responsible for what we're seeing. And the timing isn't quite right for the diatom transitions that we see, so we don't think that it's probably directly related to it, although I think we couldn't rule out it as having some influence on what we were seeing. And there's a lot of those sorts of transitions that people don't really think about, like the ecological consequences associated with them, that I feel like, they can have a huge influence on the ecology, not just fish, but any invasive species or introduced species have huge consequences on diversity of systems. So, and that's something that we regularly focus on, because I live near the Great Lakes, and so all of the Great Lakes have undergone a lot of introduced species damage, basically, associated with water quality, water clarity, and the ecology of those systems. So I think all those things are kind of interesting. And then to pick up on a little bit of what you were talking about with cave systems, there's also cave systems in southern Indiana as well, and some very spectacular cave systems. And I have an interest in cave systems as well. I had a student who was keen on the idea of collecting diatoms from cave systems, and then they asked me, do diatoms live in caves? And I was like, it doesn't seem like a place where diatoms could live. But then I started really looking into it, and anywhere there's sunlight, you can expect diatoms and other algae to sort of colonize it. I think some of them are introduced in many cases, and I read some papers about the light systems that they put into cave systems actually can give enough light that the algae can photosynthesize around it. So people, they have to be really careful about, like you said, you can bring in algae on your shoes and not realize it. And so just the light systems that run through, so that people, like touristy sort of cave systems that have walkways and get a lot of visitors, can introduce a lot of species to those systems. And I'm sure people don't think about it.
Yeah, I think that, in regards to June Lake, many places that I've studied in California and also in the Mount Rainier area, we kind of can't underestimate the impact of early mining as well. So Mona Lake was originally a mining community, so there was gold in them there hills, and that was the reason why colonization this place, because if you've ever been there, I actually sadly couldn't visit Gull Lake, because Gull Lake, at the time I was visiting the county area, was being used to fight forest fires. So Gull Lake was designated as the water reserve from the Tamarack and Dixie fires that were absolutely raging.
Good news, everyone!
So, and mining was a huge thing.
Good news, everyone!
So where Lee Vining is today in the area, I believe, was originally kind of like, I think it was called Monoville. I'll have to check the historical record. But mining was a thing, and because Mono Lake's saline, kind of a terminal lake, meaning it doesn't have outflow, fresh water was being pumped from elsewhere to actually sluice the ore. So that could be something that could also absolutely affect the intervention too, as well as introduce tons and tons of new nutrients. Mining, particularly at that time, is very mercury heavy, and I think that that's one of the concerns of Mono Lake too, is that there's also kind of naturally occurring
Good news,
saline and
everyone.
mercury and kinds of noxious chemicals at the bottom of Mono Lake, and if it dries up, it'll make these kind of like noxious windstorms that will blow over Southern California. So it wasn't completely about nature and loveliness, beauty to preserve Mono Lake. It was also about keeping people safe. So, but I think mining is something that we definitely need to consider as well. I specifically saw this in visiting springs and seeps in the Mojave Desert. Almost
Good
all
news,
of them are
everyone.
associated with a mining site. So, and you can definitely see why that would be. So, and I think that there was such a huge diversity between springs, and I think a lot of that was due to the mining activity or the camping activity. I also found, also were especially in the desert where there's water, there's life. So I found clandestine drug labs, and I also found people living out in the Mojave off the grid. So, which was an interesting thing to do by yourself in the middle of the desert, 10 miles from the highway. So.
While we were chatting about anthropogenic systems, it looks like my friend OpenSet has raided our channel and brought in their viewers, a huge clump of people. And
Good
so
news,
I've
everyone.
been sort of focused on trying to get the SEM working and listening and talking to my guest, listed on the
Good
the
news,
little talk
everyone.
bubbles as StudioCornix. Shelley's for people who are just coming in, she's a scientist and an artist.
Good news,
and
everyone.
also a software programmer. Is that what you said you do in your day job? And so
Yep.
she's sent me these samples that she collected from June Lake, and we've been working in June Lake for the last couple of years, and well, more than a couple of years, but doing research there on diatoms.
Hey,
And so,
Maori Souri.
hey, thank you for the subscription, Robert. And so I have some of them on the SEM, and then we're also just kind of like having a chit-chat time between me and Studio, and PacificPlankton's also hanging out quietly in the background of the chat, occasionally piping up.
I'm here. It's been really fascinating listening to you both. I don't want to interrupt. It's very cool. I'm like nodding along, going, yeah, yes.
So thank you for the raid, OpenSet, and I see that PacificPlankton's taken on her moderator activities there and giving you a shout out. And thanks for people who are making comments. Where is June Lake was asked by Boba Binks. So June Lake is in California, sort of near the border with Nevada. So if you know where Mono Lake is, you might not know where Mono Lake is either, but it's kind of like a really large lake in that system, in that basin. And I don't know, you could probably give a better
Nothing is
description of people from
impossible.
California
Not
than
if you can
the
imagine
guy from Indiana telling them where it is.
Yeah, June Lake is, it's in the Sierra Nevada mountain range. It's about at 7,000 feet elevation, I think, so it's up there. It is near a small town called Lee Vining, California. It's along California Highway 395, I believe, so that's kind of the extremely picturesque highway. It is very close to the Nevada border, so I would, it's kind of like it's a basin area that kind of goes into the Great Basin Desert, so kind of beyond. So it would be kind of, if you're thinking in your brain, it's going to be closer to Carson City, Nevada, than it is to any place in California. So.
And then I got a question from a viewer here, Martine Den, 41. They want to know if I have open access data sets. And they're working on neural network that differentiates different microscopic species and then paints them with different colors. I have a student who's graduating this semester, a PhD student, Hung, who was trying to use these tools, the sort of GIS tools that people use for neural networks, and trying to use object recognition software, and I don't have any open data sets that I can give people, but if you're just looking for like a set of diatom images, like light microscope images, I definitely have a ton of those, and I could probably help you out. What I'd recommend is that you join the Discord, and then we can talk about that sort of like when we're not in the middle of a bunch of other things. But that's a good question. You beat me to it, Pacific. To talk about. So I'm interested in that idea, and my student tried to do some stuff with it, but then he got busy trying to actually do his work for his dissertation, and he never really got back to the question. We were going to try to see if we could use the object recognition software to sort of detect slight differences in species from either SEM images or from light microscope images. So I definitely have a huge library of diatom images in both light microscope and SEM stuff, though, and I'd be happy to sort of share it with somebody for science purposes, as needed. So the diatom that we're looking at right now in the SEM, this is the genus is Anumastus, and the species is Rostratum, I believe. I guess it might be a different one, but I think that's the one that I remember seeing the most in that system. There's just a couple of species of Anumastus that occur in the western part of the U.S., so it's like one of two or three, but it's probably Rostratum. And this is an internal view, so you're looking, in the scanning electron microscope, we're basically seeing things in 3 dimensions, but we're using electrons rather than light to see them. And diatoms sort of are people like to liken them to a petri dish, or younger viewers sometimes say a pokeball. But the idea is that the 2 pieces sort of fit together. And we're looking at the inside view, like the diatom has broken open, and now one half of it is basically laying in the valve view so that we can see the valve face, and we're just looking at the inside view of it. So the scanning electron microscope lets us see all kinds of amazing detail, of course, and we're looking at current magnification is around 8,000 times. So a typical light microscope bottoms out at 1,000 times, typically somewhere in that range, and then the scanning electron microscope basically just goes until things get blurry for us. So on this microscope, we've managed to get something like 200,000 times and still collected an image that I thought was reasonable. Actually, that was PacificPlankton's. When she was here, she was trying to push it as far as she could. I think she got a 230,000 times magnification image, and then I just ran it through a little topaz and cleaned up the blurriness and the noise, and it looked like a normal image. So not bad. Sorry. So what was the previous diatom? It was a Stephanodiscus that we looked at before this. It was an internal view, and I'm not actually totally sure which Stephanodiscus it is. It may have been Caruscus. I didn't see any valve face photoportula on it, and there was a piece of junk on there that looked like it could be, but it didn't have the really strong costi between the striae. So Anna's asking questions. She's an actual another diatomist who regularly visits the channel, and so her questions are a little bit more specific um, than the sort of general conversation. I'm going to zoom in for people who are here. I just want to look kind of at the one end right here. Anomestis have really spectacular internal structure and external structure. They have these really sort of complex system of striae and areolae on the valve face. And then this is the raphe. This little feature down here that looks like a rolled tongue is the halictic glossa. And then on the outside edge of this, it has what's called a pseudosepta. So it's got like a little lip on both ends of the apex, where there's a pseudosepta sort of sticking out. So it's a really nice, clean view. One of the things that's nice about collecting live samples or modern samples, or getting them, I guess, in this case, is that you get some cleaner images. Sometimes stuff that's been in the sediment record is a little bit ratty or dissolved, and depending on what the water chemistry is like. But this sample is actually nice and pristine. Most of the diatoms that are here, if they're not covered with a bit of clay, are actually in really good shape. And so we can see all kinds of really interesting structural components as well. If we sort of zoom in, we can see some of the details of the areolae and their pore covers down in here. It looked like we could see them anyway. So you'll note sometimes it looks like the screen is kind of fuzzy or blurry. That's because I changed the speed settings when I'm trying to get things into focus. If I need to move around, then I usually will change it. But you can see, I think on these, you can see here these little pore coverings. So it's got like sort of a clover shape, like a four-leaf clover shape to it. And then there's some really intricate little pore coverings over the surfaces of these, which I think are sort of spectacular. And then you can see that there's different sized and shaped areolae associated with the outside edge and the axial area, and then there's sort of like a row in here, these like butterfly shaped ones that are a little bit different. And that's the sort of spectacular detail you can see in a scanning electron microscope that you'd never see in a light microscope. In a light microscope, these just look like little dots. And we're at 47,000 times or whatever right now magnification, so that actually adds a lot to the clarity of our image to be able to zoom in that closely and still resolve things clearly. So.
Yeah, I'm really excited to see this level of detail. That's obviously a scanning electron microscope to refer to. So
I don't own it either. The university owns it, but it is in my lab. So I have exclusive, unlimited access to it if I want. And Corkboard said that their partner just walked in the room and says, "Wow," every time I'm watching you. You know, that's what we're aiming for, is to try to get people to see the detail of diatoms and the beauty of these microscopic organisms. I sort of wish we could look at some of the live cells with this sort of detail. Usually, in order to get them clean, I have to put them in acid or hydrogen peroxide to get rid of all the organic-y parts. And what we're left behind is just the rigid skeleton component, which looks great in the scanning electron microscope. But a lot of times there's, you know, we don't have any color. We don't know any of the other organisms that were present in these systems. And so it's great if I want to just look at the diatoms. But, you know, Studio's specialization is like all kinds of algae. And my processing, step one is like get rid of all that other junk, except for the diatoms, basically. So hopefully you weren't expecting me to put a bunch of the green algae or cyanobacteria in the SEM. We probably could do that. We probably can make some samples that have that. I just would need to tweak the SEM settings a little bit. And it's a little bit harder to rinse out live samples and get all of the organic sort of stuff out of it without losing the actual organisms. So.
Yeah, also, I found in my collections, generally, how I collect is I will collect more towards the top of the water column, and I tend to sample from algal mat. So things that you can kind of physically see a colony happening. And I primarily also work with freshwater too. But I found that the filament algae and cyanobacteria actually decay very quickly. So if they're not in their specific environment and you don't immediately kind of culture them to get them into the right conditions, they kind of turn into goop really quickly. So within like a couple of days or weeks, depending on what they are. So I try to look at a sample as soon after as I can after I collect it to capture those organisms. I think that maybe a dry specimen preparation, so kind of like the traditional old school, like scoop out the algae, dry it out on a card, might be a little bit more conceivable because you could actually coat those and look at them. But there's also the desiccation. So one of the beautiful things about diatoms is they've got a skeleton, right? So they've got bones essentially, and we can look at the bones. Whereas the taking water out of the cell structure of cyanobacteria and filamentous algae and green algae can really be detrimental to the to being able to observe the cell structure and being able to observe the organisms.
Yeah, usually to prep those and things like tardigrades, water bears, and rotifers, and things that basically don't have a skeleton. Usually to prep them for the scanning electron microscope, you have to sort of like silicify their skeletons. So you have to do this really complicated chemical preparation technique that like takes the skeleton, well, the outer wall of the organism, whether it's multicellular or single-celled, and and crystallizes it basically with silica, and then you can put it in the scanning electron microscope and they don't collapse. But I usually say,
Good
oh,
news,
when you
everyone.
put something in there that's like an algae, that's not a diatom, or like if it's a rotifer or a copepod or whatever, you end up usually getting sort of like police outlines of objects, you know, just
Good
like
news,
the
everyone.
ghost of what was there, like, or they look like, I like to say they look like the lawn ornaments that people have at Christmas
Good
time.
news,
when
everyone.
they turn off the pump and then they sort of just turn into a deflated trash bag with some of the characteristics of like a snowman. That's sort of the way that they look in the scanning electron microscope. I have been able to get, so my koi pond, I have a little koi pond in my backyard that I sometimes collect samples from and bring them in and put them on the SEM and whatever. I have been able to get some pretty good images of Cenedesmus on the SEM because they come straight out of my koi pond, and then they're a little bit more like rigidy structures. The Cenedesmus and well, all the desmids basically have a little bit more of like a rigidity to their system. So if I don't put them through processing, like if I don't put them in hydrogen peroxide or nitric acid, and I just sort of dry them as long as I can kind of get them, you know, so they're not in giant clumps, they actually do okay. Pediastrum and Cenedesmus, and I think we've gotten some other things that we could clearly see. Sometimes they even make it through processing, but yeah, they a lot of times they end up looking sort of like sad deflated algae, as you called them. And then the question is, can you, so Corkboard asks, can they survive a vacuum? And, well, we don't usually try to put the living organisms in there. I don't want to be cruel, but this SEM has a low vacuum mode, and you could probably put stuff in there, just it wouldn't be it would charge up probably if it was wet. So it's going to be dry, whatever it is. And diatoms can survive in those conditions, so we probably could get diatoms that have like drier, you know, like assisted state. but but I don't think that you would see anything better. You'd probably see things worse because the little bit of organic stuff that's coating the diatoms would make it so you can't see these sort of beautiful ultrastructure components that we're looking at now. So yeah, you can. They might be able to live through it if I didn't coat the samples in gold. And then the other thing I wanted to say was you mentioned how a lot of the green algae and the cyanobacteria just turn into goo after a little bit. And I've been working on some cryptobiotic soils, and we managed to get some samples out of those where we were looking at some algae and a lot of lichen actually, and but they still do kind of like turn into kind of goo piles with a little bit of structure. So I have some cool images from that I'm going to probably share in the next couple of weeks, and as the sort of art displays for those go forward. But diatoms are surprisingly robust, and the samples that I got from white sands, when we first got them, I tried to make some SEM stubs from them, and then I put them in the refrigerator, and then I pulled them out after about 4 weeks of being in the refrigerator and put them on the light microscope, and the diatoms just came right back to life. Like in the light of the microscope, they started crawling around again, and I was like, wow, like that was a solid month in the refrigerator. And they'd been shipped to me from New Mexico, and I mean, they came with cold packs and stuff, so they were always kind of chilled, but they came right back to life. There was living diatoms crawling around on my slides, and I was like, that's crazy. Like a solid month in the refrigerator in total darkness, and they didn't seem to be bothered by it at all. Yeah, they're kind of show-offs, right? They've got all the good, cool ultrastructure, and they can sort of handle all kinds of crazy extreme conditions, and you know, whatever.
Yeah, so as I kind of mentioned, these samples are going to be different because they're sampled kind of the top of the water column. I did also send what I labeled as sediment, so that was sampled in the bottom of the lake bed, but not too deep. So it would have been about 3 to 5 meters, I forget which one, out off of the swimming beach in June Lake. One of the really cool things about June Lake is that there's like hummus floating. It has this like very volcanic, like taking the cure spa like vibe to it. It has like the water is slick because of the minerals in it, so it's really like a very lovely place to go swimming. But I also wanted to see kind of specifically what was living at the bottom because there's kind of this extremely fine siltiness to it. But how do you actually get your samples? I know that your sampling style is incredibly different from this.
So, not too different from your approaches. Actually, we have some stuff that's collected with plankton nets. So that's sort of the approach that PacificPlankton uses for collecting her samples, or I guess it's exclusively the system she uses for collecting samples that she shows from San Francisco Bay. And then a lot of times our samples are collected from, well, if it's collected from a core, we actually have like either a metal tube or a polycarbonate tube that gets pushed into the sediment, and then we have the whole core that comes out, and we just chop it up like it's a piece of cheesecake into samples that are the size, the resolution that we're looking at, and then those are just processed with either hydrogen peroxide or nitric acid, typically just hydrogen peroxide, to get the diatoms so that we can extract them. But if we're collecting from live material, I also sometimes will, so plankton nets, I have a what I call a macro pipette, which is just a turkey baster that I use to, I will just pipette like a little bit with the turkey baster off of the very surface of mud if I want to get an epipelic sample, if I want to get something that's living on the mud surface. That's an approach that I regularly will use. So in my kit, my things that I take to the field with me is usually a turkey baster, a plankton net, um, a ladle, or a like a I don't know, like a soup spoon sort of size thing that you could just scoop some mud with. And then a lot of times either a toothbrush or like a paint scraper to scrape off rocks. Because there's a lot of diatoms that live on rock surfaces, on mud
Daddy
surfaces,
is cute,
and
Daddy
then
is
on
strong,
plant surfaces.
Daddy
So
is
for I
brave,
actually use a
and
pair of
Daddy
scissors
will
usually
not
for that. I'll just chop up like the plant, or I'll take leaves from plants and then just put those in a Ziploc bag, for example. So the thing is, um, I don't care whether the organisms live or not. For most of the time, I'm just like, well, I'm going to find what diatoms were living here anyway. So I will usually try to look at it live, but I don't do any sort of special, like, I'm going to preserve it kind of approaches. Sometimes people use Lugal's solution or ethyl alcohol to try to preserve the living cells. But I'm usually just trying to get at the skeletons, so I don't usually care whether things ultimately live through that process, and I just usually ignore the rest of the algae, or I try to get rid of it when I go to look at the diatoms, because that's my focus, obviously. So I don't spend much time sort of like worried about those cyanobacteria or green algae. But when you collect plankton, you always collect everything. And as you mentioned, the cyanobacteria in particular they have sort of like gas bags of flotation tools to stay at the top of the water column. They kind of have either gas in their spines, if they have little spines that stick off that help them float, or they just make like fatty oils that basically allow them to stay at the surface, especially when they're reproducing, and stay in the sunlight. So diatoms don't usually have a lot of those features, so they tend to be a little bit farther down in the water column. So we might, use a plankton. My plankton net's usually got a weight on the bottom of it to carry it down, and then I do vertical tows most commonly, unless I'm near shore. So mine's a little bit different than, like, I know Pacific just sort of throws hers out in the water and then lets the current sort of carry it. And we use mine like that when we're looking at river samples. The current's usually strong enough that the weight won't even hold it down. It'll just sort of be pulled out tight on the rope. But most commonly I use it as a vertical tow to try to get some of the diatoms that might be in the deeper chlorophyll maximum areas. And you mentioned that you had some samples that were sediment samples, and actually those are also processed. We've been looking at the floaty part, and these are the sediment ones, so I just switched us over to take a look at it. There's no reason why I had to just sort of sit on one sample the whole time. I always prep like way more samples than I could possibly look at in a 1 or 2 hour stream. But these were processed the same way, so we just put these into nitric acid and then on a hot plate for about 2 or 3 hours. They're supposed to be boiling, but we don't actually ever boil them. We just sort of make it warm because we have things to do. We don't want to worry about the samples being like completely dried out. But as a result, I think we sometimes get a little bit more organic matter in ours than people usually want. But I just want to get the stuff that's attached to the diatoms off of it, basically.
Yeah, that's interesting. I just always kind of wonder how folks do their sampling, because I feel like I don't have any equipment that I bring with me. I do have a kind of a scooper, essentially. It's essentially like a pool cleaner, but for science. So it's like a net that I will bring with me. Those are what I use generally to collect the cyanobacteria and algae from algae blooms, so that I can drain out some of the water. Because in order to get a color from your algae bloom, you need to lyse the cell, so break up the cell walls. And as you mentioned, diatoms specifically, I don't usually extract pigment from diatoms because they are so tough. They also, certain pigments are soluble in certain kind of solutions. So a lot of the green algae, which have kind of the carotenoids, those specific accessory pigments will extract an alcohol, but for diatoms, they have a, you mentioned it yesterday, they have a specific accessory pigment, but what I've seen in my literature is that it's like most readily dissolvable in things like hexane, things that I am like not gonna touch. So I usually use either actually moonshine or methylated spirits. So I will go to the liquor store and buy like the cheapest rot gut, like 195 proof corn liquor that I can, and use that to extract the accessory pigments. But things from bacteria, those are phycobilins, so those are a different type, those are a whole different chemicals. They're actually even a different like evolutionary branch of photosynthesis, which I love. Those generally are water soluble, so I'll use like phosphate for saline, or I'll just use like a salting out process. So yeah, what is that that specific kind of pigment that are that diatoms have that other algae doesn't? Remind me of that.
I think they have one that people just call like diatoxanthin, and then there's fucoxanthin, which I think are the two like usually only occur in like the golden brown algae or the golden algae or whatever you want to call diatoms.
Yeah, fucoxanthin. Yeah, and
I think
there are,
I was going to say the fucoxanthin, I think, is also in the macroalgae and the larger brown ones.
Yeah. It's a sort of brown algal pigment, and then the diatoms have their own that's like diatoxanthin or something like that. I'm not a pigment person, so but I think that I've seen that before, and I thought, oh, well, we know where that one comes from, right? So it's got the name in it.
yeah, fucoxanthin is definitely in the larger algae species. I think it's commonly a lot of these pigments are sold in aquaculture. So people who raise fishes or hermit crabs, you can feed them certain things to achieve colors, right? It's kind of that phenomena. Flamingos are pink because they eat the brine shrimp that eat the algae that have the red pigment in them.
Yeah.
So I know that fucoxanthin is it's sold as all different kinds of like health remedies. I am suspicious of all of them, but it's another antioxidant. I think that one of the things that like aside from like aquaculture, a lot of the algae products that kind of go into cosmetics, food products are antioxidant properties. So which jury is out on if or anything. I mean, for example, like spirulina is healthful. It's good for you. It's nutritious, but you would have to eat it in such large quantities that I'm not sure. Like it's like it's this idea of like these organisms being a superfood. And I'm kind of dubious of that because they are traditional food sources in a lot of places. Like I think even in the Mono Basin, for example, the Paiute tribe that lived in that area really ate a lot of the things that were in Mono Lake, especially in kind of like survival times. So like survival foods would be like the brine larvae for that were present in the lake, and those feed off of the algae. So I think that like a lot of this was kind of like a supplementary food, which yeah, it's interesting. And in addition to diatoms being used for kinds of stuff too, which scarily like the toothpaste thing just freaks me out. Like they're so sharp. I would just hate to. They're still used in some tooth powders too.
Yeah.
I found this out
Yeah.
recently.
Sorry. The natural toothpaste or tooth powders will use diatoms, but they're tiny, so for our scale, they probably would barely notice them, their sharpness. I think. But they ate sea monkeys. Is that what you're telling me? The sea monkeys that I bought when I was a little kid and we grew up in an aquarium.
Yeah, you can actually eat them. I did while I was at Mona Lake. My friend ate some of the brine shrimp larvae out there. They're salty. They were, again, like a survival field food, so something where if there were just other food sources not local, you can eat them. Yeah, and yes, they are essentially sea monkeys. I didn't have the heart to include any in my samples, even though they really wanted to come with a sample. I decided to leave the brine shrimp and everybody out there in Mona Lake. Although there were some pretty cool algae blooms on the side of the lake.
OpenSet said they tried to look at toothpaste under the microscope looking for diatom pieces, but they couldn't find any, and they were using Arm & Hammer toothpaste. Probably they just use non-diatom silt in those. I know there's silt, there's silt-sized particles. That's why diatoms are useful for things like toothpaste, because they want something that's abrasive, but not something that you're likely to spit out of your mouth like sand. And a lot of times, because I'm trained as a geologist, and we try to explain to students that in order to tell the difference between silt and sand and clay, you just or like a shale, for example, or a siltstone, you rub it on your teeth, and they're like, oh, like, why would you rub it on your teeth? It's like, well, that's, your teeth are sensitive to grit size, basically. Probably more sensitive than anything else that you have when you get down to grit that small.
That's horrifying.
It's like licking rocks, biting rocks, chewing rocks. I don't know. I think that's a level of dedication that I don't have.
You don't want to put it in your mouth? You don't want to see what it tastes like? That's what people always ask me. What do the diatoms taste like? And you know what? We have somebody here who actually tastes different algae, right? Like, occasionally. Don't you eat or make things out of algae? Didn't you? You had some recipe.
I made one of her drinks, actually two. I've tried both. They're beautiful, lovely. And then the snacks, so cool. Yeah.
Yeah, I mean, you can, like, a lot of, well, first of all, let's go ahead and we'll just put this out here. There are things that are eatable and there are things that are edible, right? So that is a nuance that we should appreciate. So most green algae is eatable. You can eat it. It will not harm you. Just the way that wet newspaper, you can eat it. But being edible, actually tasty, is kind of a different thing. But yeah, like, you can definitely eat a lot of different kinds of microalgae, and we obviously benefit from things that further up the food chain that eat it as well. I've never tried diatoms. I've only used diatomaceous earth in, like, gardening kind of context, because the microcuts can harm insects, right? So it's also a lot of times used in treatment of bed bug infestations too. So people will put, like, cups of diatomaceous earth, like, around their beds, so that if the bed bugs try to crawl, they'll crawl through, and the silica bodies will, like, cut them, which will interfere with their feeding and eating. So there's definitely use for it. I have no idea how a diatom would taste. The way that I describe to other artists, because I actually get the same question for some reason as people. We're like, okay, what does it do for me? How does it taste? Can it make me beautiful, right? And you're like, yes. But the kind of, like, I say, I'm like, well, imagine that you had a Kit Kat bar, only it was made of glass. So you bit down on the glass, and the Kit Kat bar, instead of delicious nougat, was filled with chlorophyll and pond water. And they're like, okay, cool. So that's kind of how I would describe it. It's like you took a little test beaker and you broke it, and it was full of, like, like the juice from your green smoothie, only with no good stuff in it, just spinach. So.
That's not how I usually answer that question. When people ask me what they taste like, I go, "Oh, they taste like Smarties." And I've never tasted a diatom, but I imagine they taste like Smarties. They look like candy. So, but then I tell them not to eat them. You know, they're the Smarties you shouldn't eat. But yes, I can't imagine that they're very tasty. And I try to usually tell people that they're too small to taste. You know, like you'd have a hard time tasting something that was as small as a single diatom. But maybe if you got enough of them together, you know, you could put it in your mouth and eat it. I really like your distinction between between edible and eatable. Pacific Plankton wants me to put a mustache on this diatom, apparently. So I will do that. Let's see. We have mustache technology.
I was hoping for the beard, actually.
I didn't bring the beard from home.
Oh, man. Okay.
But
Shucks.
distract people for a minute and I'll get it. I'll see if I can get it from
Distract
online.
people.
Yeah. Just to talk about something cool with Michelle here, and
Random question. Well, there was an interesting question in chat about farming the seas and how kelp can feed the world. And I always thought that was an interesting idea. And when I look at the oceans, I think about how it's the way we're using the oceans the way land a long time ago. And as we are able to use the oceans more industrially, I'm curious how well we'll be able to manage the resource as opposed to the way we've managed like farming industry on land. I just think it's a really interesting, we're at an inflection point where we can go the wrong way really fast. Or maybe we already have. I don't know.
Yeah, I think that, so I'm very interested. Kind of one of the things that I kind of see in my future role, I in my kind of day job, I make the user experience for software, the software structure, how software works. But I see this as coming into the kind of future ideas, and this is kind of an emerging practice of biodesign. So actually kind of designing things that will have biological nature in the future. And I think that this brings up a really interesting question. I try not to be a pessimist or about it, but we've done a very poor job of trying to biodesign in the past. So we've thought things have been a really good idea. So for example, natural fencing, and that's how we get things like multifloral rose in Midwestern United States or Himalayan blackberry here in the Pacific Northwest. I also want to caution too that, you know, like we bring our own food cultures with us, and a United States and kind of Western centered food culture has a lot of different cultural and like historical precedents to it, including like heavily things like the earth being our domain, in addition to telling people what they should and shouldn't eat, which I am very, very hesitant to do in any sort of context, because a lot of folk rely on fishing. That's their traditional food source. For example, like the Duwamish people here in the Seattle area, who are yet an unrecognized tribe, relied on salmon and other kind of food sources that we traditionally have, like as like we've got to eat less of this. This has to be farmed. Whereas, like, you know, this is a very traditional and incredibly sustainable way of living. So it's really interesting, and I think that that's kind of like algae is sold as these kind of like biodesign future solutions to everything, right? Which, first of all, puts a lot of pressure on algae, and secondly, I kind of am skeptical, but they're sold as these superfoods. Like if you eat spirulina, it has all these wonderful qualities to them, right? But like it's also like in what quantities and how we actually make that, you know, a good practice, whereas it is healthful. And cultures that ate spirulina did have health benefits from it, but that was kind of like a specific context. Also, I hear a lot of times, like, especially when I'm speaking to artists about like the plastic digesting algae and the plastic digesting bacteria. And I was like, so imagine a future where all of your plastics are magically in peril. And that includes things like the plastic parts that have been body surgically. It includes medical plastics. It includes like industrial plastics that hold bridges together. Imagine that. So, we think of it as this positive, wonderful thing that cleans up an oil spill. We never think of it as slowly rotting our houses or our bodies. So, it's an interesting thing. I think that it's like, I think that there's a huge potential there, obviously, but I think that it's also something that we can't be too utopian about, especially like kelp fields too. Those have knock-on effects. Anytime we grow something for our own benefit, like there's stuff that happens. Like what happens if we do suddenly have like massive kelp farms on the coast? It's the same thing with oyster farms. Like, is it a coincidence that we farm oysters but have like a higher incidence of red tide? Maybe, maybe it's climate change, maybe it's acidification, maybe it's harmful algae blooms, maybe it's many, many millions more of these individuals producing a toxin that may have not been produced in such quantity. Maybe it's all those things. So anyway, that's
it's fascinating. I just think we're at such an inflection point, and I really love that beard and those googly eyes. Thank you.
It's you spent your channel points on it, so hopefully it's.
I did.
Very
satisfying.
Grab a screenshot of that, and then we can put it in the Discord.
Go ahead. Well, I'll grab it from the replay later. I'll clip it.
Okay. So I'm gonna do away with it now. We're gonna hide it. It's hilarious to me to have a really serious philosophical conversation while I'm in the background drawing a smiley face on a diatom.
I feel like that's how the internet works, right?
It's exactly how the internet works. Perfect encapsulation of the internet experience.
I'm curious too. So this is my collection and not yours.
Mm-hmm.
Are you seeing the same things that you usually see in June Lake samples? Very curious about that.
So this is the diatom that I described. This is Stephanodiscus caruscus right here, and it's a common plankton species in the lake system, and it usually has this sort of ratty appearance in the today system. And then these are sponge spicules, not diatoms, but they're all over in these samples. And you said you collected this from the beach, which is sort of on the, I want to say, northeast corner of the lake, right?
Correct. Yes.
And
It's
then,
Pacific is O Ridge campground, so that would be if you want to do a lot of longitude later. That's where it's at.
yeah. So, and then this is the external view of that Anumastis rostrata that we saw the internal view before. So I took a little picture of that early on while we were talking. That is a diatom that I'm used to seeing in that system. And then that little guy that we took a picture of that had the beard was Lindavia costii, which is another plankton that shows up in the lake in the more recent stuff, but is one that we commonly see. So I'm seeing a lot of the same species of diatoms that we usually see. This is Epithemia, I want to say Sorex, but I'm not positive because I'm looking at it from the bottom. But I think it's Epithemia Sorex, which we saw in the samples kind of a lot. So all the regular, usual suspects are present. And then I think that's a chrysophyte cyst. Yeah, so there's a chrysophyte cyst. So most of the diatoms that I'm seeing are the same ones that we see, but I want to add a slight caveat to that and say that my view of what's in the lake has also been biased by the fact that you collect this about maybe 3 weeks after the most recent collections that we did from June Lake. So the modern samples that my graduate student Laura collected for us from June Lake, just like I said, just a few weeks before you were out there. So I suspect that it's going to look the same to me or look pretty similar. And the samples that I have that we collected from the surface of June Lake, which is similar to what you have here, that I had from before were collected 2017. So they're not very far from in time from what your samples were in terms of when they were collected. And then this is that same stuff that we saw early on when we were looking around at stuff from I think on the other stub. It's maybe Niagara or like Niagara, I'm not sure, but it's in that group. And we see this appearing sort of with humans in maybe a little before they show up, but in that sort of like industrialized era, this diatom starts to show up in the core, and then in the modern records we see this present. But if you go back in time, that's not a diatom that we typically found in the lake system. And then there's a lot of this cockanies as well, which is just stuff that they live on macrophytes or submerged plants. So those are all over the place, and there's bits and pieces of those in here as well, and they were all over in your other sample. It's just a common, I think they were Cockanies pediculus, which is pretty common, although maybe we see the other one, lineata, a little bit more frequently in samples that I've looked at. So, but I haven't actually analyzed the samples that Laura collected from this summer. That's on my to-do list to actually finish our collection from June, so that we have a paper we're working on that's pretty close to being published, where we're looking at the distribution of diatoms and then trying to relate that to where would be the best place to core in the lake, based on what kind of diatoms are preserved in which sections. So if we wanted to get basically a thermal stratification signal out of the lake, the core that we collected is actually perfectly located for that. But if we wanted to actually get a lake level reconstruction, which is the other thing that we want to do, our other core that was collected and hasn't been analyzed yet would actually be the ideal location for that. So I feel like after we did this analysis, I kind of feel like we need to analyze that the other core that we collected from this lake to try to get a better sense of lake level change. And I think we're going to try to campaign to get a student who's interested in paleolimnology to analyze those for us. So I'm probably going to put out some advertisement for master's students or whatever who might be interested in that project. And then, as I mentioned, we're going to write a grant, or we're trying to write a grant right now to look at the longer history and look at diatoms and how they may, as some of the diatoms may have evolved in the basin. So that's on my to-do list, to try to get that grant together before I think it has a March deadline for the program that we're targeting. So, and it'll probably be the same suspects involved in the grant writing, me and Mike McGlue, and then my friend Trisha, who's a sort of a diatom geneticist. So we're going to try to get some ancient DNA from the sites, and then we're going to see if we can link that together with the evolution of the diatom that we actually see in the June Lake record. So it'll be a really interesting proposal, and I'm hoping that it'll get funded and we'll be able to sort of explore that topic a little bit more in detail. But that's drifted off from your actual question. But yes, most of the diatoms we're seeing are the same ones that I've seen from June Lake in the past.
How often do you mentioned that your most recent core was very close to when I collected this summer, and then previously 2017. How often do you actually go and sample? Do you resample?
Do you know what? I've never been to June Lake, despite working on it all this time, because Mike went out and collected the samples before I was actually involved on the project. and then he got a student, and they were looking for a project, and then she was interested in diatoms. So like my involvement in the original core collection was didn't happen until after they'd already collected the cores, and then they went back out to do some modern sampling in the summer, I think, of 2019, and I was on sabbatical in that summer, so I wasn't actually available to go out with them. And then COVID happened, and then they went out this summer, but I had a ton of students that I was working on in the lab with, and so I also didn't go out this summer. But Mike has gone out to the Sierras to look at those materials and to collect stuff from those lakes, I think on 4 separate occasions. And they only collected cores from June Lake one time. So I think both of the cores that we have are from 2017. And then when they went out this summer, they collected a whole set of cores, but they actually collected them from Gull Lake. So they just collected a few modern samples from June Lake from the shoreline. So probably not dissimilar from the sample that you have here with respect to like how and where it was collected. But when they collected, I think it was the modern samples were 2019. They or 2018. They went on a boat. They had a grid, and they basically had a grid of sites that they collected from. And they did the same thing this time when they analyzed the Gull Lake samples. They collected the cores, and they did like a grid of surface samples from the bottom of the lake from a bunch of different sites. And then Laura went and collected some of the modern samples from living organisms from the meadow area, and then from some sites in shallow water where they could walk around in hip waders and swimsuits or whatever and collect their samples.
Cool. I'm planning to revisit sites that I visited this year because the Dixie fire was so huge and changed the landscape. So there were actually places where driving to the area, I couldn't drive back the same way because of fire. And I'm very interested in kind of the way that forest fire affects algae, specifically the way we also treat forest fire. We, here in the United States, Canada, and I know Australia as well, use Phosgene, which is the fire suppressant. So that pink stuff that you see being dropped from planes, helicopters, that is actually a proprietary blend of fugitive pigments and ammonium phosphate. So it's actually fertilizer, and it's been shown to directly link to algae. So I'm very interested in kind of seeing, like, not only that, algae, like you said, diatoms are super tough. Algae is incredibly resilient, cyanobacteria especially so they're kind of the pioneer species. They're those things you see in the cryptobiotic soil because that's the only thing that grows there after a volcano erupts or you have a blowing saline desert out in the middle of the day. But I'm also kind of interested in how the environment really change or affected. I would love to see if like next summer's June Lake like poppin' with diatoms because of all of the fire activity in the area. So
this is actually, it's funny that you would bring this up. This is actually an area of research that my lab actually does. So in 2016 or 2015, I had a student here, Sabrina Brown, who now has a PhD. She did an interview with us one time on the stream as well. So she went to Nebraska after she finished here, and now she has a job at Defiance College. And her master's thesis was looking at the diatom response to fire. And so she collected charcoal and diatoms from the same core. And then sedimentary charcoal will give you a sense of whether it's a long distance fire or a close fire based on whether it's large particles of charcoal or little tiny pieces of charcoal. And then she was looking for a direct interaction between the fire and how it might affect diatoms. And this wasn't from June Lake, it was from Island Lake, which is in the Beartooth Mountains. And so that paper came out earlier, I guess it came out in the January of 2020, where we looked at the results from that finally got published. And then I think she had a subsequent paper where we talked a little bit more about it last year that came out, or maybe it was earlier this year, and looked at the longer record. And so it's one of the interesting stories that came from that. It wasn't that people were using fire suppressants to fertilize the lake, but rather in these systems what we saw was that the fires basically devastated the shelter belts that are around the lakes, and so wind would start to affect the lake much more strongly. And then we actually saw the response from the diatoms that could do better under windy conditions. That would be a direct response to the fire. So we would see a fire, and then we would see these sort of wind specialist diatoms that like the lake to be more mixed show up for about the immediate response was within basically 5 or 10 years of the fire, and then basically you wouldn't see as much of a response again. So we thought that that was associated with a combination of the fire breaking the shelter belts, and then also fires, as I'm sure you're aware currently, people in the West Coast are particularly aware of the fact that fires are usually associated with landslides and debris flows, because they take all the vegetation off the landscape. And so that usually means there's a giant pulse of sediments that come into lake systems. And so that affects water clarity, and it has a whole bunch of effects on stratification and potentially effects on the total dissolved organic carbon that comes into a lake system. And so it has all these really complicated potential effects associated with it. But I wanted to mention that my current student, Laura, who has collected the cores and is looking at the diatoms from Gull Lake, is looking at the fire history of the basin by we collected charcoal from each of from one set of the cores, we collected diatoms and charcoal. And so we'll have a fire, she's almost done analyzing actually the fire history part. We just don't have an age model for the cores yet. So we can't do like accumulation rates, but we can actually look at like particles and peaks in both macro and micro charcoal particles. And then she's also analyzing the diatoms from those cores. So we should be able to see if there's a direct link between the fire history and the diatom response. But all that stuff was captured before the really big fires that came through this year. So it would be kind of interesting to see what sort of response that we would get next year. So that's actually would be really interesting. And I'm going to guess that Mike will go back out next summer, because he has continuing work that's going on in the Mono Basin, and so he'll probably continue to do some research associated with it. And maybe we should talk him into either taking me along or taking Laura along, or maybe I can just lend him my plankton net, and he can sort of see what's come out of the plankton this season as a response to that. It would be really interesting to see.
Yeah, first of all, you should go to June, because it is quite beautiful. It really is, and it's at least when I was there in late July, it was wonderful to swim in, and was just quite lovely. But yeah, I think that I am using forest fire charcoal in my artwork. I'm also using it as kind of loose charcoal. The kind of one of the kind of conceptual pieces of my after Ehrenberg work is that in the composition there's kind of like a rondelle around a round form. I'm sorry, confined, but they kind of emerge out of a smoky kind of background, and it's kind of the idea is emerging after the cycle fire. My current that I have is Lion's Head fire in Oregon, so it actually completely destroyed the community of Detroit, Oregon. I believe it's in the Willamette National Forest. but I do want to collect charcoal specifically from the Dixie and the bootleg and the camera fires to incorporate into future artwork. But it really is kind of a concern too, because part of fire is very natural. So I was living in Washington D.C. for a while, and one of the things that I read while I was there is in New York City, when it was New Amsterdam Dutch colony, Dutch people, like, there was like an annual burning of the forest, and basically it was a big party where you get on boats, sail down the Hudson, drink beer, and watch the fire burn. And it was a controlled burn that would actually burn the forests and underbrush. And anyone who's been in the eastern United States, the forests there are intensely brushy underneath. They're incredibly brushy. And that's kind of the, even though there aren't these massive, truly massive, super hot fires, really, east of the Mississippi, it's another kind of showing of how there have been fire intervention happening in the United States for thousands and thousands of years that we stopped or did different things to really change the forests. It's very interesting to me to actually see that kind of that history in the work reflected. Like you're saying, like 150 years ago, stuff started changing. What was it? Was it mining? Was it farming? Was it draining the aquifer? Was it introducing more nutrients? Was it the timber industry? What was it? So it's very interesting to me, like, to kind of try to narrow down those specific things.
Yeah, I think sometimes it's difficult to disentangle them because they either point in opposite directions or sometimes they point in the same directions. And so a lot of times what we're left with is just trying to figure out the timing. And even that can be challenging because their responses to things don't usually happen immediately in nature. It's usually some sort of a lag, and then the time lag is like a challenge for us to figure out, like, like when is the gap, which one of those gaps is relevant, and then also our ability to date things accurately to like a year is pretty small typically, unless you're looking at tree rings or ice cores or something. You don't usually have that sort of resolution. It's usually like plus or minus 10 years. That's a reasonable age model for a lot of things that we analyze. And so we don't usually have great records of stuff that allow us to get that sort of a resolution out of it to go, well, this is definitely this thing. So there's a lot of sort of inference that's left, and you have to make a decision about which one of those things you think was likely to be most impactful. And you mentioned things like mining, which probably have a profound influence on lots of aquatic systems. But, it's sometimes difficult to figure out, like, okay, there's a bunch of cadmium or something gets released as a result of this mining. So, like, how do diatoms respond to cadmium? And the answer is, I don't even know that people know the answer to that very well. So a lot of heavy metals, we know that they can basically distort their skeletons when they're building them, if it's in the water. But aside from that, I don't know that we know that diatoms have like a specific response to particular heavy metals, or we don't know what they are, other than just sometimes it can be bad, right? So I think that's a part of the challenge, is like trying to sort out both the timing of events, and also like sometimes the effects that are there that are potential drivers don't affect every part of the ecosystem the same way. And a lot of the other organisms aren't preserved in the record, so diatoms have an advantage for us, and that they're very powerful tools for backing out environmental change, but they don't respond to everything, right? And sometimes it's difficult to get at that, what would be a good tool to get at mining, for example, where diatoms might not be the best tool. But I don't know what tool we have, so I think it may be a tool we either haven't discovered yet, or maybe there's just some things that we just don't have a great way of ever sort of backing out exactly what's the root for that.
Okay, quick question. I'm going to butt in. Anna wants to know what this is. She keeps asking, and I just wanted to make sure I got an answer. She's been really patient.
It's Epithemia frickia, I believe, for Anna. I believe that's a epithemia for sure, and I think that's frickia. I think that's the species. But I haven't done like I think I just called it Frikia and didn't bother to do like a look at the striation or any of those other things, because it just has the shape of Frikia. And so I think I need to, this is one of those that was like a question mark. I just had it as like Frikia with a question mark after it. So I think it's likely Frikia, but if you don't agree with that, I will not take the criticism as like something that I want to argue about. It's definitely epidemia, though. My guess is that it was Frikia. Does that satisfy her curiosity, hopefully? So
Hopefully.
one of the things I wanted to do was also showcase some of Studio's work. And probably the easiest way for you to do that is just to put some links. Did you already do some of that? I can't see chat very easily while I'm running the SEM, but maybe you could put some stuff in and then talk about it a little. If it's something where I can actually get the image, I can put it up on the screen. But if it's like Instagram, I can't really like show Instagram easily. So.
Gotcha. How would it be best to provide you images? I have a bunch.
Just throw them into Discord, just to send it to me in Discord, and then I can just paste it right in. So that will work perfectly, and we already have a thing there. You can just paste it to me as a private message in there, and then it won't be exposed to people to download, other than me. So I know you said you did some charcoal stuff. I remember earlier in the summer you were working on like a card set, like a playing card set that looked like it had some charcoal. Is that one of the things that you did with the charcoal?
The playing card set was part of a art exchange. So what it was is it was actually I was exchanging. We all had a deck of playing cards that we were doing. Most of us are some sort of artist. So those were not done with charcoal. Those were done with Sumi ink. So, but again, it was kind of like I've been thinking a lot about forest fires and a lot about kind of the emergence of organisms after forest fires. So this one is part of the Ehrenberg series, and it's not oriented otherwise, but whatever. If you turn it clockwise 90 degrees, it'll be in the correct orientation. If you can't do that, it's fine. But you can see the charcoal in the background. The blue that you're seeing in this particular piece is spirulina, so it's spirulina extract. It's a watercolor that I made from spirulina pigment. Some of it is from I extracted farm. Some of it is industrially provided. But this is really fashioned after Dr. Ehrenberg's historical record of organisms. So what I did is took a sample in this particular location. This is at the Oregon River, the Oregon Trail crossing of the Deschutes River on the Columbia River. So this is someplace where the Fremont expedition would have likely camped. This was also a place described by Lewis Clark and a traditional kind of place where the Warm Springs and Nez Perce folks would meet to trade, as well as the Walla Walla Indians. So this is a place that I suspected was a very likely sample site for what was actually provided to Dr. Ehrenberg. And it actually bore out like scarily well. Like I saw so many things that were so similar. But again, I've seen a lot of variants in Pinolaria that were not actually in the original. They are nowhere to be found in the original finds. So it was really kind of an interesting thing to see, like to just go back and take a look, see where I suspected that the sample was from. So
It doesn't let me rotate it. It just automatically rotated it to this landscape view. So sorry about that.
It's on my site and through my Instagram. So yeah, but this is kind of just retracing those steps. And again, thinking a lot. I think that like what I'm so excited about is that there's like really strict scientific inquiry about the things that I'm curious about too. So here at the Deschutes River, this is a place that had incredible importance, cultural importance, as well as human intervention. So there's now a highway, there's now the railroad, there's like a railroad bridge across this area. There's 2 state parks on both sides of the mouth of the river, as well as the community of the Dalles. If anyone played the Oregon Trail computer game as a kid like I did, the Dalles, that's where you float down the Columbia River to the Dalles. So this was a place that was like, has been really important. It's had human activity. It's part of these various expeditions, but it's also someplace that has had a huge amount of change. And one of the things that I love so much about kind of visiting Ehrenberg collection is that a lot of these samples were taken before the pre-industrialization of the area. So we're talking about like 1840, between 1840 and 1870. So this is before Portland, Oregon was a place. This was before Seattle, Washington was really a place, before the establishment. Like all of these samples are labeled Oregon, and that means Oregon Territory, which at this time would have been from Northern California all the way up to kind of where we see as Alaska. So even in parts of British Columbia, us American, United States folks and Canadians were still like fighting like little petty skirmishes about the borders over like stolen pigs and like jars of brandy and things like that. We'd be like, "You took my brandy," and then pop off a few shots and eventually settled on the folks at Parallel, I believe. So, but this is really interesting to just see like what the interventions are, because this is one of the largest agricultural sections in the United States. So famous Washington apples and cherries and peaches and onions and wheat come from here. So it's also like it's this huge part of the land that has had intervention. So this is another one. Apparently all of these are going to come through sideways, but that's fine. So this is the Columbia River at what was Fort Walla Walla, so near modern-day Wallula, Washington, so also known as Walla. And this is one of the very, very few colonial settlements in the area. And I believe that this particular sample was collected by the United States Exploring Expedition. So this expedition went all over the world, but they really intensely mapped Oregon Territory and went through the Puget Sound and the Strait of Juan de Fuca to and down the Columbia River to what was the Whitman Mission, which is today Walla Washington. So again, exploring kind of the like intense agricultural and population intervention using a sample that used to be like that when it was taken was not very populous.
So for most of these diatoms that you have in your drawings, I can identify them easily to genus and sometimes to species.
And that's the thing too, is that like I wanted these, like, this is kind of like generally my work is not super so there's what I wanted to do is specifically kind of show that reflect the way that Dr. Ehrenberg would have approached this work, which is beyond just being able to describe things and talk about them or write about them. You actually want to have a visual kind of representation of what these particular organisms are, because this can really give you an idea. In addition to, like, I mean, this is obviously not as good as a scanning microscope image or a light microscope image or something that could be, we tend to trust photography. This is like a cultural phenomenon, which I find very interesting. An illustration of a crime scene would not be as good in court or permitted at all as like video surveillance footage, even though an illustration could have a lot more fidelity than some grainy gas station video surveillance. But for some reason, we tried an image. But I also kind of do want this to be part of like a historical record of what this place was. Like here, Fort Nez Perce has a very weird, like very U.S. colonial-centric like placard and explanation on it, and it even uses the term "the white man." Yikes! So it has like this very like Eurocentric kind of like message to it, but it's across from Wallula, which I believe has 300 residents in it, and then it's just a placard along the railroad track. So the fort is completely gone. The site is completely gone. So this was actually sampled in the Walla Walla River at the confluence of the Columbia River in the McNary Wildlife Refuge. So part of this is also kind of retracing the historical record and just seeing like a lot of stuff is gone. It's just gone. Or it's on private property. That was a lot of what I saw in California too. So I'll grab some of the card projects too. So the playing card project.
Yeah, these are really cool. I really, really like the watercolor. And I should point out, of course, that I do a lot of scanning electron microscope work, and last night we did a little, what I thought was going to be a little stream of me showing people like how I colorize SEM images, and I of course do drawings of diatoms as well, not fancy watercolor stuff. I'm actually, of all of my skill sets, I've never learned to paint, but drawing I can do, and digital drawing and rendering are like things that I'm actually much more comfortable with. So I do a lot of like diatom based art, but never anything with painting. I can make stuff look like it's painted. That's about it.
Yeah, I definitely like the painterly direction that you have been going in for sure. I also think what's really interesting is that, like, when I show folks who are new to scanning electron microscopy, or microscopy, I'm sorry, I always say that wrong. When I actually show folks that, the first thing I hear is like, is this the actual color? Like, is this real? Does it really look like this? So I do think that that's really interesting too, that there are lots of reasons why we manipulate these images or colorize them, not just for beauty's sake, but also to, as you pointed out in your stream yesterday, emphasize the internal kind of texture or structures. And it can be like, but, you know, there's an interesting thing that we trust that image, even though it is a false color. It's not real. It's not true. So.
Yeah, well, and that is a question that people ask me occasionally as well. Like, this one also came in sideways, huh? It's just what it does. Sorry. Turn your head sideways, people.
Yeah, it's your old Windows. It can't handle it.
Probably. There's some things it's very good with, like running a scanning electron microscope, though.
This is OS, which grumble. Anyway.
Your aces are all desmids.
Yes, the aces are all desmids. I love desmids so much. They have a special place in my heart, mainly because of their evolutionary link to land. So I think that's so cool. I also think that desmids, just in their form, can really show you how cells were striving to be multicellular organisms, in addition to their just general beauty. So these are done in gold leafing pen, and each one of them is just bicycle playing cards that I used black gesso to prime the surface, because these are like impregnated with like silicone or something, so they're really hard to paint on. And then I used Sumi ink to kind of simulate a forest fire look to them. So they're all gold. Some of them have like a but yeah. So this is an entire deck of playing cards. So each suit kind of had a different thing. Generally, they reflected the number rather than the organisms, but the aces are desmids for sure.
Cool. So you did every number, not just the face cards?
I did. Yep, I did every number. So it was a full deck of cards that I'll be exchanging. They'll basically be shuffled together with the other artists, and we'll each get a full deck of cards back with a variety of different arts. This is through an artist residency program that I assist with. So just last week I was assisting with, it's the Ayatana artist research program. So it helps connect artists with scientists to expand artistic practice. And our emphasis was infinitesimal, so it was all things small. So we speak to folks who are involved with physics, chemistry, just all kinds of things of like examining the small.
Very, very cool. I love that idea. It's like exchanging Pokemon cards with somebody, but science Pokemon cards, right? Which is
Right.
actually the way that I like to think of diatoms. There's just so many of them, and they all have such different shapes that they're a little bit like Pokemon. You know, you need to like keep looking for the really rare ones, right?
Yes, yeah. I mean, I'm a big player, so it's really not surprising that I talk too. And Pokemon was actually developed because through the kind of hobby of bug and insect collecting in Japan. So that's actually what inspired the original Pokemon game. So it was this kind of like naturalist like pursuit. So it's not surprising that they're so similar. So
I'm a little too old for Pokemon in that it came out after I was in college. Hey, is this one actually came out like oriented correctly?
this is. I think I need to like export them. Like they need to do some sort of like JPEG like export project that I haven't done some Pokemons. So this is Sanguina. This uses algae pigments primarily, and it's fading over time. So this is a portrait of the that I was talking about, and it's done in anaxanthin, anaxanthin. Perhaps it's the red accessory pigment. It's done in the yellow that is extracted from Chlorella vulgaris. The blue comes from spirulina. The orange comes from Daniela salina, and the green also comes from Chlorella. This is also mixed with watercolor, and I've exposed it to light, and it's just slowly kind of fading. So I can grab it off my wall here so you can see how much it's faded. So. Oh, no, you can't, because you can't see my... Never mind.
I can see you. They can't.
Well,
I can see it.
So
oh,
you
yeah.
can see
Yeah. It's fading just a little bit. The blues especially.
Carotenoids are particularly fugitive. So this is kind of a step documentation of making the painting and then documenting it over time as it fades. So I think that kind of like conceptually talks about like the changes of a microbiome over time and the cycle of bloom and bust that algae go through. So
this is a super cool painting. And is there... So you want it to fade? Is there a way you could preserve it if you put some sort of shellac over it, or?
I haven't found a way. So I've tried. Everything has a UV coating, so everything is like a UV coating. I've also tried BPA, which is a food preservative, to make these a little bit fast. But I think that a lot of people out in the algae pigment industry are trying to figure this out too, but I haven't figured out a way for it to like be more light fast yet. So that's a couple of chemistry iterations in the future, I think.
Just you have to... It's probably oxidizing, is my guess, is part of it, right? So it's probably that'd be very difficult to keep it from doing that in a porous medium that's painted on the surface, right? So
yeah. I mean, a lot of these are again, for like the sources for these are food or cosmetics, so they're not preservative products. So, like they have preservatives in them, but they're not meant to be legacy products, paintings. And I also even think that the concept that your pigments will last forever is kind of presumptive and also very modern too, because we have things like verdigris, which was a really popular green color used in Renaissance oil paintings that oxidizes over time, turns brown. So we have these like very dark brown sort of paintings that were actually bright green back in the day. So there are, and we also see these changes regularly kinds of historical artwork. So like brilliant tomb paintings in Egypt that just simply by people walking through and breathing on them went away. So
Okay, well, I actually have to end the stream soon because my daughter has we have to go talk to her teacher about her grades or something. It's like once a year we get to talk to our teacher about our daughter's grades. And so I need to get back to the house at 3 o'clock or my wife will kill me. So,
do it.
But this has been really awesome. It's really nice to see your work and to be able to share it with people. And thank you to Pacific for both moderating the chat, which I have a hard time managing while I'm also doing the SEM and interviewing people. And then thanks to OpenSet for the big raid for bringing people in. It's very nice. I think we've had 40 to 50 people on the channel for a large part of it as a result of that. And thanks to people who've had questions and helped us carry on the conversation a little bit. I really like to do interview streams. That's actually one of my favorite ways to sort of be on Twitch because other people have to do a lot of the talking instead of just me. So that's makes my life a little bit easier, but I should probably try to do it while I'm not also trying to run the SEM. So, but I think it actually is nice to be able to showcase like your art and also the samples that you sent me and some of the diatoms that are present in those samples, and then to have a nice conversation about June Lake because it's something that actually I can be conversant in, having studied it for years now. So we should probably find somebody to raid. I got a sense from talking to you that you were thinking about streaming. Is that still something that you're thinking about doing?
Yeah, I need to get some upgrades in my technology setup. I've only used my light microscope for reference, so I don't usually use it for its photographic integrity, but it's starting to like extremely me. But in the future, I do plan. I'll probably, let me say, like I'm strongly thinking about it. So that would be a light kind of stream, as well as possibly like the drawing, so the actual illustration of the organisms themselves. I often work from, so yeah.
That would be really cool. Really neat to be able to see both the microscope and the sort of artistic connection to it. And I think it would be really cool also if you just did like some of your paintings as streaming. I think it would be really cool for people to see that and to sort of interact with you on that level. Just throwing out possible suggestions. One of the things about streaming is once you start doing it, then kind of like it spirals out of control if you let it. So I try to just sort of do some little things every once in a while that are different from the main thing that I'm doing, which is mostly looking at diatoms on the SEM. So, and then I don't have any great idea for who we should raid. Maybe I've got this Paul Pape Designs that is doing art. That would be fun. Or Vicenia Designs is also another artist that might be a good choice, since how we're coming out of sciencey art. Or we could go raid a Rocket Roundup talk show from CosmoQuest. So I'm gonna force Studio to give us a suggestion. What do you think we should raid? A science and art?
I think everyone always appreciates it. So, but let's raid an art. I think that
Right.
We'll have kind of like a transition into artistic aspect.
Okay, let's see what Paul is doing. I'm gonna just click on it really quick and see if I can get, I gotta mute so I don't get commercials. But I'll see what they're up to. Oh,
Yeah, don't get commercials. We don't want commercials.
they're sculpting Mii characters from Nintendo Wii.
Ooh.
Sculpting them out of what? I don't know yet. But that might be okay. And then my other option is Vicenia. She's a artist that draws stuff. So you can choose if you want, whether you want to see sculpted Mii drawings or sculpted Mii art or whatever Vicenia is up to. Actually, she's doing some really cool, like, hand-drawn marker art. You have a preference?
Let's draw marker art.
Let's do the marker art? That's great. Okay, so we'll do Vicenia. Vicenia is part of a group of people on Twitch. She was in the Artist Back Alley, which is a group that they sort of have people, Miss Daisy D and Little Chook, who are artists who hang out in my chat frequently, asked me to be a part of because I do sort of sciencey, arty stuff. And then on the weekends, what they do is just raid from one channel to the next. It's really great. Once a month, they just have all the artists basically get one little 1 hour time slots, and then people just go from one to the next to the next to the next. So if you're thinking about checking out other art streams, you might see when those are, and I'll probably advertise the next one when it comes around. And Vicenia was one of the people who did the first raid train with me when I was involved with it. And so she does really, really cool drawing. But I think just one note is I think she probably, so I tend to be very family friendly, and she tends to just say whatever she wants. So keep that in mind, that there might be not so family friendly stuff. But, you know, her drawings right now look really spectacular, what I can see, what she's working on. So that'll be great, and we'll just end nice right on time at 2:30 Eastern time, and you guys can get about your day. I want to again thank Studio for taking time to join us. This has been a lot of fun for me. Hopefully you've had some fun. I wish I could have gotten the video aspect functioning, but you know, Windows 7. But sometime in the future, maybe we'll do another one. Cool.
Thank you so much, and have a wonderful day.
Yeah, this has been great. And now we're going to go raid Vicenia. We'll take a look. We'll raid an art, which is, you know, one of the things we like to do is raid music and art. So we'll see everybody next time, probably Friday afternoon with Sylvia drawing. And then we'll see you then.